for joining us again for the HPL seminar. Today, it is my pleasure to introduce Dr. Jackie Lebenzon, an assistant professor in the Department of Biological Sciences at the University of Calgary. Dr. Lebenzon completed her BSc and PhD at Western University, where her doctoral research was focused on the mechanisms of diapause and cold tolerance in Colorado potato beetle. She then held an NSERC postdoctoral fellowship in the Department of Integrative Biology at the University of California, working with Dr. Carolyn Williams to investigate the mechanisms of flight muscle remodeling in winged dimorphic field crickets. Outside the lab, Dr. Lebenzon is a hobby potter and also enjoys creating insect-themed ceramics, bringing her love of insects into both her science and her art. Today, she will be presenting her talk titled Mechanisms and Consequences of Flight Muscle Remodeling in Insects, in which she will explore how insects dynamically remodel flight muscles and the physiological and life history consequences of this process. So please join me in welcoming Dr. Jackie Lebenzon.
Great, thank you so much. Can everybody hear me okay? Okay, awesome. Thank you so much for having me. So I've been at UCalgary for almost 2 years, a year and a half-ish. And I've been wanting to meet people here for so long, so I'm glad this is a good excuse to do it. So as Asha said, I'm Jackie. And today I'm going to tell you a little bit about the work that I've done kind of throughout my career, my PhD, during my postdoc, and a little bit of what my lab is just starting to do. We are new, so we haven't gotten much done, but it is still very exciting. So before I start, I just want to acknowledge all of the traditional lands that this work was done on and where I lived over the past 10 years. I think that's really important. And I also just want to thank my teams. None of this could have been possible without my team Beetle and team Cricket. Lots of folks from Western, the USDA, Cornell, of course my lab here at Calgary, as well as my collaborators in the US at Berkeley, University of Louisville, and William and Mary. So yeah, people are great. And I will say a lot of this work was done by undergrads, and I will highlight them as I go. Okay, so obviously I'm an insect environmental physiologist. I'm in the comparative physiology community, so I love insects, but I also love all sorts of other animals. But I know you guys care about humans. So I will start off with the statement that I think we can all agree on, that vertebrate muscle remodeling is usually defined by growth and maintenance of muscle, right? I don't look like this, maybe one day, probably not, but I definitely try and maintain and grow my muscle as best I can so that when I get older, I can, not fall and be somewhat fit. But the problem is that, muscle atrophy is usually maladaptive. So we're trying to grow and maintain our muscles so we can avoid muscle atrophy. It's maladaptive in the sense of aging or diseases like sarcopenia, where we're losing muscle mass. Maladaptive in disease contexts like cachexia with cancer. Obviously maladaptive in neurodegenerative disorders like ALS. And of course, the elusive space travel. If we want to get to Mars, we need to be able to know how to, avoid this atrophy that a lot of astronauts experience in microgravity on board the space station. Like our astronaut here that basically has to spin in class for, hours a day just to maintain and maybe lose a little bit less muscle mass. Now these are humans. So muscle atrophy is usually maladaptive in humans, but not in insects. And so I'm going to tell you a story today about what I like to say is adaptive muscle breakdown. And there's at least 5 orders of insects. I don't know if you've never heard of these names before. These are 5 orders of insects which we know adaptively break down their muscle. And I say adaptively because it's not from disuse. There's not really anything to do with disuse atrophy in insects. They can maintain their muscle really well. Instead, a lot of them will adaptively break it down as a natural part of their life cycle for some sort of physiological reason. And I'm going to quickly convince you why I think insects are cool and why we should study them. Well, first of all, there's a lot of them, right? In this, I really like this image. It shows, half of terrestrial animals make up, our arthropods. There's lots of termites, lots of ants, lots of other arthropods, which I think in total, it's like 75% of all terrestrial animals are bugs. So get used to them. So it's really important to understand this diversity. Insects can help us understand a lot of fundamental processes. I'll highlight this and, say this is mostly Drosophila, but Drosophila are bugs too. And a lot of really fundamental discoveries we've discovered in Drosophila, like the Nobel Prize for the toll pathway, or the molecular mechanisms of circadian clocks, and even the discovery of heat shock proteins in, the 60s. That was in insects. And so I think we can learn a lot from them in terms of fundamental processes. Of course, here we're talking about muscle. And the most important thing is that insect flight muscle is analogous to vertebrate striated muscle. So here is an electron micrograph from one of my colleagues of a deer mouse gastroc muscle. You can see myofibrils here at cross section and all these dark, swirly, happy mitochondria. While a beetle muscle looks basically the same, albeit a little bit more organized, I will add. But it's the same. They have the same structure, they're neurogenic, and so it's a really nice model to maybe better understand muscles in general and not just about insects. And I think what's really interesting is, you know, I'm a physiologist, I'm a metabolic physiologist at heart, I'm really interested in energy metabolism. And we know that insect flight muscle remodeling is really tightly linked to energetic stress. And so today I'm going to tell you two stories. First, I'm going to tell you a story, and I'm just going to give you the spoiler alerts now, that hibernating beetles can adaptively degrade their flight muscle to save energy and then regrow it all on demand when they need to fly in the spring. And we're going to talk a little bit about that. We're also going to talk about crickets. And we know that these crickets selectively degrade one muscle type while maintaining a neighboring muscle in response to what we call flight for reproduction trade-offs. So in ecological physiology, we're always really interested in how animals allocate energetic resources to different processes. And the big one is that flying is really expensive and so is reproducing. And so it's really exciting to kind of tie in muscle remodeling to that. Okay. So we're going to go over or at least I'm going to try and convince you that learning a little bit about these extreme but natural examples of muscle remodeling can actually teach us about the mechanisms, the consequences, and to some extent the limits of striated muscle. First we're going to talk about the beetles and how they remodel their flight muscle to maintain energy homeostasis during the winter. And then we'll talk a little bit about the crickets and talk about how they can actually remodel their muscle to reallocate resources between life history strategies like being flight capable or having babies. Okay. So why do we care about winter? This is kind of the basis of my research program now. And I think we can all agree winter sucks. Chinooks suck a little less, but today it's very cold. And that's what makes winter the ultimate challenge. In the summer, it might look like this. You might have a bug happy on a leaf eating, it's warm, it's balmy. As soon as winter hits, it's going to be really cold, but there's also going to be no food because most insects eat plant life or other insects, and both of those are kind of seemingly gone over the winter. But of course, I've spent most of my career studying why this doesn't matter because insects are actually really good at surviving the winter. We can talk a little bit more about that in the question period. Even though you don't see any bugs outside right now, I promise you they are everywhere. Maybe you don't want to hear that, but they're everywhere, I swear. And they're really good at surviving the winter because they can enter a state of dormancy or hibernation that we call diapause, and that's what I'm going to use throughout this talk. And it's kind of analogous to what you would see in a bear hibernating or a tiny ground squirrel, but insects do it too, and they're just as good at it, if not better. And a hallmark of diapause that we know is that they massively suppress their metabolism, and we as humans can't really do that. If we knew how to do that, then we could really space travel. But they massively suppress their metabolic rate so that they can save energy. And then what's even cooler is they can reverse it really quickly in the spring. So they can do really expensive things like fly and mate. And for context, flying insects, their resting metabolic rate basically goes from 0 to 100-fold higher than that when they initiate flight. So they're really metabolically flexible. And what we look at in my lab are these diapausing Colorado potato beetles. We have a colony of them in my lab, and we think they're a really great model for studying these types of things. They're a great model for a number of reasons. I think they're pretty cute. You can disagree with me. But they're an agricultural pest. And so people care about them. I always joke, people care about how to kill them, but my lab is trying to study how they survive. So, there's tension there. But overall, they live in potato fields. So this is an image taken by a graduate student who's here that this summer they were collecting some beetles. These are just potato plants, potato farms in southern Alberta. The adult stage and all of the larvae live on the plants. They lay their eggs on the plants. They do everything. They decimate plants. And so farmers do not like these, which is why they call us to come and collect them when they see them. And we can bring them back into the lab. And what's nice is they can be pretty much like lab rats, A good model is one that you can rear and you can induce things in the lab. And so what we do is we bring them into the lab and we use kind of day length as a cue for them. Just like, August hits, the days start to get shorter, we hate ourselves and, winter's coming, blah, blah, blah. These beetles can also sense that. So we can expose some of them to these long day conditions and basically catfish them into thinking it's summer all the time, and they're very happy and they breed and and they live out their best lives, but we can also expose them to short day conditions, and that makes them think that winter's coming. What we can then do is put them in little bins filled with soil, and after about 9 weeks, they'll enter hibernation or go into diapause underneath the soil. And if we're patient enough, we can wait around and they'll pop right back up. And that's kind of their winter simulated life cycle. And I started showing this video to show people that they're very cute and charismatic, but this is them in their like artificial diapause bins. You can see these guys are starting to bury a little bit. These guys maybe are starting to dig down. And they like to overwinter in the soil where it's a little bit more buffered than if they were to just be hanging out top the ground. And this gives us a really nice model to be able to then, sample at various time points throughout this process and understand what's going on. And of course, they're a great model for metabolic expression because they do indeed suppress their metabolism. So if we take these beetles and we put them in a respirometer, we can measure their carbon dioxide production as a proxy for metabolic rate. And you can see that in diapause, they suppress their metabolism by about 90%, so they're operating very low levels of metabolism. And then they reverse that really quickly when they emerge. And I want to point out something that's just lots of cool things about these beetles. This metabolic rate was measured before they've even started to eat. So they have this flexibility where they suppress and go back up before they even eat anything. And of course, they degrade their muscle. And I say this casually, but really, you can't, I don't know if you can see this date. The first paper that showed this was in the 60s. And then nobody ever talked or spoke of it again. And I was doing my PhD trying to figure out something cool to do. And I think I came across this paper and was like, oh, this is really weird. And then I found some more papers that randomly cited it. And they're like, oh, well, this probably, lowers the cost of their maintenance. If you get rid of muscle, it's probably a good idea. But at the same time, I was hanging out with a lab that were a bunch of mitochondrial physiologists. And I was like, I like mitochondria. I want to hang out with them. And then I realized that insect flight muscle is very, very mitochondrially dense. The cell is about 40% mitochondria. And so that kind of led me down this rabbit hole of wait, maybe it's not muscle atrophy. Maybe it's not disuse. Maybe they're doing this for a reason to save energy. And then there were a couple other hints that we figured out that it's not just muscle wasting. So this is us showing that it's adaptive muscle breakdown. It's not just atrophy. We did some RNA-seq, and to simplify it, we found that within the flight muscle, they show a lot of differential expression of transcripts related to what we call mitochondrial homeostasis. So a lot of genes that are involved in making new mitochondria, breaking some down, changing the structure, changing the function. If this is just, an atrophying, not used muscle, why would we see this much dynamic activity going on in the cell? And finally, I threw some permeabilized muscle in a mitochondrial respirometer, and we found that the oxygen consumption of the individual mitochondria goes down almost to nothing, just like their whole animal metabolic rate did. And so all of this was supposed to be a side project. This turned into my whole PhD because I was like, it's not just muscle wasting. This is so cool. They're doing something weird. And so it kind of led me to ask the question, if it's not just muscle wasting, then how are these flight muscle mitochondria actually contributing to metabolic suppression? And so I did, what all biologists like to do. We actually like looking at cells, And so I used electron microscopy to look at some of these flight muscle cells. And here you're looking at a non-diapausing beetle muscle, diapausing beetle muscle, and right off the bat, they look very different, right? Mitochondria are obviously here, surrounding this really nice myofibril, but in diapause, it looks very different. I want to point out the key thing. I know this kind of looks like gobbledygook to people, but I want to point out these weird structures. These are called trachea or tracheoles. Insects don't have lungs. They breathe through tiny tubes that go from the outside of their body straight into their cell. And if the muscle were atrophying, why do they need oxygen? And why are these tubes still inside of the cell? And so that's another really good point, that they're not just, getting rid and wasting away, but they're actually just remodeling what's inside of the cell. And then, of course, when you let them come out of diapause, everything is back. They've regrown the entire mitochondrial network. All the myofibrils are back, all before they've eaten anything. And so this is pretty impressive degradation and regrowth that we're seeing. And they do this all while maintaining their nuclear integrity. So I don't know if you can see, these are just dappy stained cross sections of the muscle. And they have identical numbers of nuclei. This is multinucleated muscle. And despite the inside looking like that, they still have that many nuclei because they need to, be able to make stuff and keep the cell running, right? And this is where I get to be excited and talk about work with my current students, because my current undergrad Zoe is also finding that they suppress apoptosis. So if we measure a marker for apoptosis or caspase 9, we see as they enter diapause, it's actually suppressed. So a lot of people, oh, they're just killing all their cells. They are not. They are getting rid of little bits inside, but not totally getting rid of them. And if you take a closer look at like the whole process, this is on the bottom here is kind of just a time frame as they go into diapause, where the end here is kind of when they're deep in it. We start to see lots of lysosomes, which are important to break down parts of the cell. We see some really sad, pathetic, there's a single mitochondrion. By 9 weeks, you've seen this already, everything's gone. We see these multilamellar bodies, these really loopy structures, which are basically remnants of membranes from structures called autophagosomes. And we can see autolysosomes, which are also really important for recycling material. And if you zoom up, you can see like broken down bits of stuff in mitochondria inside, which is really cool. And then you can also catch another important thing for cell recycling, which is these double membrane autophagosomes. This is all to say that, we found our answer. Metabolism seems to be suppressed, all the way down to the mitochondria level. And, it's actually just driven because they're just taking a hammer to all of their muscle mitochondria, which is very strange because typically you want mitochondria because they're good. But in this case, it's adaptive to get rid of them all. So that led me down a rabbit hole to think, okay, well, what's actually breaking down the mitochondria, right? And that is where I really started thinking about the concept of mitochondrial homeostasis. It's really important in all cells, all eukaryotic cells, anybody that has mitochondria in their cells. You need to be able to get rid of damaged mitochondria and make new ones. And so there's a pathway called mitophagy or mitochondria-specific autophagy, which removes mitochondria. And there's a bunch of different pathways, but what I focused on in my PhD was the pink one, parkin pathway. And all you need to know is these are two proteins which tag mitochondria for removal when they're damaged. And this kind of starts a cascade of other cellular events that initiate autophagosome formation by proteins like ATG5, which can then fuse with the lysosome, those, heavy stained structures I showed you earlier, to create this autolysosome, which then secretes all sorts of enzymes to break down what's inside. On the other hand, if you want to make new mitochondria, you just undergo mitochondrial biogenesis. And there's, again, these are complex processes, but I've distilled them down because there were some targets very interested, and that was PGC1 alpha and NERV1, which can drive basically transcription and translation of nuclear encoded mitochondrial genes to lead to more mitochondria. And I want to add that all these pathways are conserved. So, this is happening in beetles, but this is also happening to some extent in our cells. So what we set to look at first was whether or not mitophagy is activated. Obviously mitochondria being degraded. And so this was an undergrad I worked with at Western. And we looked at the gene expression of a couple of these players. I'm just going to talk about parkin and ATG. So on the y-axis here, we have like the level of gene expression or transcript abundance. And on the x-axis is just, again, kind of non-diapausing beetles and then them going into diapause. And with parkin, we didn't see much. But by 9 weeks, kind of when they're deep in it, there's increased expression of parkin, which is sustained throughout. And then we also see this big increase in ATG5. And so it was really clear to us that our mitophagy is being activated and to some extent overactivated because, it was going nuts and degrading all of these mitochondria, not just a few. And so, I've been showing that, I guess I didn't really describe this framework, but we can think about, as metabolic rate decreases throughout the timing of diapause, what's happening. And so we're starting to build this conceptual model of what muscle is doing during this breakdown and why it matters. And so we know that mitochondria are tagged by parkin for degradation and then disposed of in these autophagosomes. And I want to add, I don't know how many of you are doing molecular work or qPCR and gene expression. We do a lot of gene expression work in insects, mostly because antibodies are really hard to get to react with insect proteins because they're all raised against mammals. But we can easily do qPCR. But so then there's a lot of speculation saying, well, it's just correlation, just gene expression. In this case, we were seeing an increase in expression of the gene that encodes the protein, which literally makes the structure that we saw at 15 weeks. So that was really exciting. We could bypass the protein issue. Okay, so this, again, it's still all correlative. So we wanted to figure out if mitophagy is actually driving mitochondrial degradation. And the only way to do that is to start manipulating stuff. And the nice thing about insects, especially these beetles, is there's a lot of genetic tools, just like in Drosophila, to be able to knock genes down and manipulate gene expression. And so that's exactly what we did. And so this was work done by undergrad Peter with me at Western. And just as a reminder, we know that Parkin expression peaks kind of just before 9 weeks. And so Peter thought, well, if we knock it down at 7 weeks, maybe this will prevent or delay mitophagy, and maybe we might be able to, mess with their metabolism. And so he injected beetles with double-stranded RNA complementary to Parkin, or a negative control, GFP. And we can talk more about this later, but basically, anytime you introduce double-stranded RNA into, well, really any cell or insect, it's going to trigger the RNAi response and reduce the expression of that target gene. And so that's exactly what Peter found, is that after 5 days, he got about an 80% knockdown in the gene expression of Parkin, and like a beautiful, almost full protein knockdown. That was one antibody we got to work, because we got it designed specifically for the beetle. And so now we had this really cool tool that we could use to then figure out if, how important Parkin really is to this whole process. And the cool thing is we found that knocking down Parkin did recover mitochondrial abundance, or almost delayed the breakdown, because we see here on the x-axis, the beetles injected with double-stranded Parkin had about 4-fold higher mitochondrial density compared to the GFPs. It wasn't quite as high as the non-diaphosphine beetles, which was to be expected. They kind of already started going into hibernation. But when you actually look at the EMs, there are mitochondria in the knockdowns and none in the other. And of course, what the coolest thing is that we were able to reverse metabolic suppression, meaning the beetles injected with double-stranded Parkin had higher carbon dioxide production rates compared to the controls, but again, not quite as high as the non-diaphosphors. But of course, science makes no sense sometimes. And although we partially recovered whole animal metabolic rate, we couldn't really detect this at the mitochondrial level. Maybe a modest increase, but not significant. And so that's just the nature of working with, non-selected lines of beetles. And so we think maybe there's still some mitochondrial suppression happening, but of course I mentioned Pink1 and Parkin aren't the only players in mitophagy, and there could be other things happening. But that's, for a conversation over beer. So basically we can conclude that mitophagy does drive, maybe doesn't regulate this mitochondrial breakdown, and ultimately metabolic suppression during diapause. Okay. And I always have people say, okay, well, the breakdown part's the boring part. What about how they regrow? Fortunately, I have to finish my PhD, so we have a little bit of data to show this, but it's kind of what my lab is focusing on now. Because, as I mentioned, kind of at this stage, the beetles are regrowing an entire cell's worth of mitochondria, rebuilding up the muscle protein. How are they doing that without eating and while, being under the soil? Well, again, we turned to some gene expression, and we looked at the expression of PGC1 and NERF, and PGC1 alpha and NERF1. And again, we have transcript abundance, or the gene expression on the y-axis, and then them kind of going into and then coming out of diapause on the x-axis. And we saw that at 12 weeks, they increased PGC1 alpha and then increased NERF1 by 100-fold. I've never seen gene expression values that high before. And so they're clearly upregulating biogenesis, but I want to point out they're still underground here. They haven't eaten, they don't really have any mitochondria to begin with, yet they're able to jumpstart the production of new mitochondria. And so we think what's happening is, they're able to jumpstart mitochondrial biogenesis in anticipation of their emergence from diapause. So for context, that would be like, you're sitting on your couch all winter, and then, April 1st, you get up and run a marathon and everything's great, right? They have this amazing capacity to grow things in their muscle without any type of exercise. So I won't go over that because I don't want to be repetitive, but now we have this really cool conceptual model that my lab is starting to kind of really test. I think a really interesting question is, how is this process cued? And I'm hoping to do some sort of ATAC-seq to see if we can find novel transcription factors that are maybe driving this. Maybe we can use that as some sort of therapy to help with, mitochondrial quality control. What we do know is that isoform switching appears to play a role. So I had an undergrad last year work through some RNA sequencing data. This is a lot. All the purple dots are basically what we call isoform switching events. So when you have an mRNA transcript, it can be spliced differently to result in 2 isoforms of the same protein. And this is happening about 350 times between non-diapausing and diapausing beetles across about 255 genes. So there's a lot of really dynamic regulation happening that could be driving this initial muscle remodel. Another big question is what's actually initiating it, because these mitochondria are seemingly totally healthy. And the hallmark of mitophagy, especially when it comes to Parkin, is it gets rid of damaged mitochondria. And I won't get into too much details here, but basically when mitochondria aren't happy, their mitochondrial membrane potential just goes to crap. And so we think that there's something else in the cell that's driving a change in mitochondrial membrane potential. And what that could be is an uncoupling protein, which you might know of in human brown adipose tissue or sometimes muscle that kind of helps dissipate gradients in the mitochondrial inner membrane, but also for thermogenesis in babies. So what is it doing in insects? Well, we know it's there because once again, Zoe's really interested in this, and she found that it's up almost 2.5-fold during diapause. And so Zoe's now on a quest to try and figure out what UCP4 is doing in an insect's muscle. So it's really interesting. And then, of course, the magic bullet question is how do they know to reverse this mitophagy and regrow? And so we're starting to look at things like AMPK signaling, which might be able to sense cellular energy status, and it might be an endogenous rhythm that is controlled by nothing other than simply time and diapause. So we're really excited to start looking into some of that stuff. Stay tuned. Okay, so I'll kind of end this story and say we found that reversible flight muscle mitophagy is what's driving metabolic suppression in these overwintering Colorado potato beetles. Okay, so I did my PhD, I'm from Calgary actually. I did my PhD in Southwestern Ontario. I studied winter for 6 years. I had enough of it, so I moved to California, study field crickets in the Santa Ynez Valley. And here, is me holding a female cricket at her field site. She has a long wing. I don't know if you can kind of see it. She's quite good at flying. And we know that because we go to catch them, and they're nocturnal. I don't know if you know, at night you hear the crickets sing, right? At night, we lay down a tarp, we put out a speaker that mimics the male crickets, and we come on ladies, we trick them into thinking that there's males there, and we can actually catch them in mid-flight, literally it's really fun. So this is my lab mate had just caught a cricket here. You can see her wing extended. Poor cricket. She did not find a date that night. But yeah, so they're really interesting because they fly using these two sets of what I call ecologically important flight muscles. And I will add that the beetles have these two sets of muscles too. I just didn't pay as much attention to them in my PhD, so we're going to focus on them for the cricket. But they have these dorsoventral muscle, which I'll call the DVM, and they go side to side, and they basically move the thorax, which then helps move the wings. But they also have some appendage, like joint. Oh God, I'm not a biomechanist. They're attached to the legs, so it can help them walk as well. Whereas the dorsal longitudinal muscle, or the DLM, we know is only used for flight. And I'll show these images, these white bits. These are the trachea, the tubes that go into the cell that help them breathe. Okay, and so the cool thing about these crickets is they need to break down their flight muscle to reach their full reproductive potential. So I mentioned earlier that there's a paradigm between allocation of resources for expensive things like flying versus reproduction, and these crickets have been studied, maybe not the species, but this order of crickets has been studied in this context for like decades in the field of ecological physiology. So this isn't that new. But we know that these flight-capable crickets, when they're flight capable, they have this really red, juicy muscle. It's weird because insects don't have myoglobin, but this muscle is very red. The beetle muscle is orange because their whole insides are also orange. But they're very red and plump and juicy. But at the same time, they have very low reproductive investment. So these are her ovaries. They're kind of small and wispy and not ready to reproduce yet. At some point in her life cycle, she's going to break down her muscle while massively growing her ovaries. And so her ovaries become something like, over half her weight. So they're just very gravid, a lot more pregnant than what humans look like. But I want to point out that what I discovered when I started in the lab is there's a point at which, and I have here, this is pink, this is white. there seems to be some sort of differential breakdown of the muscles. Okay, so PSA, I'm about to show cricket muscle. I don't know if you can see it that well with the colors here, but we photographed the muscle, and basically the side to side is the DVM, up and down is the DLM. At first, they're both red. There's this point in the breakdown where they both turn pink. But then there's also this point where the DVM stays pink, but the DLM turns white. And there's a correlation in size. So the white ones get smaller, whereas the DVMs they stay the same, they maintain. And since then, we've actually quantified all of this using literally just a stereo microscope to give red, green, blue values to these colors. So we know that it's not just a weird trick of the eye, these are changing color. And so it gives us a really unique opportunity to study this point where we have this differential breakdown. And I think it's really interesting because, obviously actively breaking down your muscle is weird to begin with, but this would be like, breaking down your bicep, but keeping your tricep around for some weird, random, useful reason. So I think it's really cool. So most of my postdoc was spent trying to answer this question. Well, how are they carrying out this selective histolysis? Muscle remodeling, sometimes we call it histolysis. How do they carry out the selective muscle remodeling to support this life history transition from being able to fly versus needing to have babies? And I took a very similar approach to my PhD where I asked, well, okay, how is muscle cell function and structure changing? And can we figure out some of these processes that are driving that? And because the lab I went to also had mitochondria things, I started there. And I was like, okay, well, maybe they're breaking down mitochondria, maybe the mitochondria are changing in a different way. So then I once again measured mitochondrial oxygen consumption rates in muscle fibers. And I guess I never mentioned, I'm doing this using high resolution respirometry with an Ouroboros oxygraph. I will have one in my lab in a few months, so if anyone wants to talk mitochondria stuff, let me know. Okay, so here we have oxygen consumption rates on the y-axis, and on the x-axis will be pretty similar throughout. We have age, so they're either 3 days old or 5 days old, and the color is corresponding to the color of the muscle that does the breaking down, or the DLM. And we have this nice age match control just to make sure that what we see is happening because of the breakdown and isn't actually because of their age. And it's kind of messy because everything is in biology, but the important thing is we can see that the gray bars are the DVM, they're maintaining the size of this muscle as well as the oxygen consumption rate. Of course, if you want to take into account how many mitochondria I used, I'm just using muscle that's permeabilized, so I don't actually know how many mitochondria are in each sample. So you can measure cytochrome C oxidase activity in this to standardize to how many mitochondria there are. And when you do that, it's all the same, which is like another boring story of, oh, they're just breaking down their mitochondria. Boo, nothing exciting. So, but it's also kind of cool because then if you look at the muscle, I again did some electron microscopy. If you look at, on the left here, my right, your left, is the DLM from a cricket with functional muscle. This is with non-functional, histolyzed, broken down muscle. Looks very different. If we zoom in a little, we can see mitochondria here, surrounding myofibrils. These are the tracheals, the little breathy tubes. This one looks very different, but I will add it looks very similar to diapause beetle muscle, which is really exciting. So they still have nuclei everywhere. They even have these tiny pathetic mitochondria that are really close to the nucleus, but not much else. This is just, these pathetic tiny mitochondria compared to these ones. And the really cool thing is if you take muscle from the same individual, this is one we're catching at that midpoint of breakdown where it has pink muscle here and white muscle in the DLM. This is the same individual. They're fully maintaining or seemingly fully maintaining this one. And this is the one that looks like it's a shell of a muscle cell. And if you put some numbers to that, again, this is mitochondrial area on top. You can see that the DVM, they actually do decrease the mitochondrial abundance. They're degrading some mitochondria, but they're stopping at a certain point. Whereas the DLM is basically none. Just those few tiny ones. But the really cool thing, which is like you don't need numbers, you can just look at the electron micrographs to know this. The DVM, the one that's maintained, they maintain mostly all of their myofibrils. So that's the functional unit of the cell, right? Where contraction is actually happening. So I think it's really interesting. They're clearly degrading their DLM, but maintaining myofibrils in some mitochondria and the other type of muscle. Okay, so we've kind of answered that question. Okay, so what processes are actually carrying these out? We've talked a lot about autophagy. We're going to talk about it some more. I'll rush through this because you guys are experts now that we know that mitochondria can be tagged. And I'll talk about protein here too, because we have quantified that protein is going down. And we can see it being, sequestered in an autophagosome, which fuses with the lysosome, which then is broken down in an autolysosome. So we hypothesized that autophagy is actually activated in flight muscle during breakdown, but that it's really this differential regulation of autophagy in the two muscle types that's leading to the selective remodeling or the selective breakdown. So how do we test this? Once again, I make fun of myself. We like qPCR. It just is really good at giving us, an overview of what's happening in a cell. And so I did a lot of qPCR because we were writing a grant to get money to do RNA-seq, but while I was there, we never got that money, so it was just qPCR, unfortunately. And so I did a lot of qPCR. I'm just going to tell you about two genes. One is called Beclin, which encodes a protein, which is similar to ATG5. It's involved in that early autophagosome formation, as well as mTOR, which might sound more familiar. It's a master regulator of everything and anything energy related in the cell. But the important thing is that it is a negative regulator of autophagy, so it should inhibit autophagy. So what did we find? Here we have transcript abundance or gene expression on the y-axis, and again, this age time frame on the y or on the x. And then the color of the bar corresponds to what's happening in the muscle. So you can see that as the DLM degrades, we see this uptick in Beclin, which is really exciting. And we actually see the same thing in the DVM, but I want to point out the differences in the axis and the axes. This one's at about 6. This one only increases to about 4. So they're both, we know that they're both, degrading mitochondria to some extent. So that makes total sense. When it comes to mTOR, if we look at the DLM, there's nothing going on whatsoever in the gene expression. But if we look at the DVM, we see an uptick kind of due to age, but also due to muscle breakdown. And so we're thinking, wow, this actually makes sense because we see that they're degrading some mitochondria. So maybe at this point, mTOR puts a stop on it and stops it before too many mitochondria are gone. So we kind of confirmed this. This is all correlative. How can we test this experimentally? Once again, we turn to RNAi because it's a really powerful tool to use in insects to manipulate gene expression. And so this work was done with one of my undergrads at Berkeley, Tomas. And he, just like the beetles, we injected crickets with double-stranded RNA complementary to Beclin, our gene of interest, or GFP as a negative control. And he confirmed after 4 days, we got about a 70% gene knockdown. And so we hypothesized that, if Beclin-mediated autophagy is really necessary for this breakdown, the knocking it down should prevent or delay any of this muscle breakdown. And that is exactly what he saw. So here we're looking at, on the y-axis, proportion of individuals that have muscle of a certain color. And at least in the dorsal longitudinal muscle, the DLM, there are significantly more red and pink individuals, meaning we've delayed muscle breakdown. But we don't see this in the DVM, which again makes total sense because we know the DVM isn't degrading as much to begin with. So that's kind of a cool proof of concept. We also wanted to make sure it wasn't just a weird Beclin thing, and so we looked at some proteins downstream of Beclin. We couldn't find Beclin antibodies. We're like, well, what antibodies can we get? First one we looked at is kind of an autophagy activator, or GABARAP-L2. It's involved in that late stage autophagosome formation. And we found that in the knockdown crickets, there was indeed lower expression, which meant we prevented autophagy activation, which is exactly what we expected. And then we also looked at cytochrome C oxidase protein expression, which often correlates to mitochondrial abundance. And of course, we saw it up in the knockdown crickets, meaning there were more mitochondria because we prevented breakdown. So it was all really exciting. And so to what extent does Beclin-mediated autophagy drive this? It absolutely appears to drive it, which is really exciting. Okay, so what about mTOR? We were thinking, how do we test mTOR? We could do more RNAi, but the problem is mTOR does so much else in the cell. We knock it down, we might mess with something else. And around that time, another Drosophila paper came out where they were injecting little flies with rapamycin. And it's just a drug, but it inhibits mTOR. And so we're like, just try and inject them with rapamycin. And we might expect that inhibiting mTOR kind of takes out the stop of the stopper. So we're we're inhibiting the negative regulator, meaning if we inhibit mTOR, then we could induce autophagy. And once again, that's exactly what we found. This is in the DVM. We inject them with PBS. Normally, none of them have any, white muscle that's breaking down. But if we speed it up with rapamycin, suddenly we start to see high rates of breakdown. If we look at COX protein abundance, we don't see much change, and that makes sense because we know in the DVM, they're maintaining their mitochondria. Whereas if we look at autophagy activation, we're actually increasing autophagy activation just like we predicted. So we're able to really show that causation and conclude that it is really that mTOR signaling promotes the maintenance of this dorsoventral muscle. I hope you're still with me. So we kind of conclude here that it's a speculin-mediated autophagy that drives selective remodeling to match these lower energetic demands. And we say that because if you remember, the muscle that's maintained is also important for walking. And if you're reproducing, you might not need to fly, but you still need to walk. You still got to find your mate. You still got to do whatever it is you have to do. So it's a really kind of interesting ecological consequence of the muscle maintenance. Okay. So throughout all this work, I always consistently got the same question. It's like, oh God, that must not be good for a cell to break down all of your mitochondria. And I agree, yes, that's probably not good. And at the same time, in the crickets, we see this really nice color transition from red to pink to white. And since they don't have myoglobin, what's initiating that transition? What's the color? And then we thought really hard and we thought, iron. Iron makes things red. Because iron is really important in, interacting with heme compounds, for example, electron transport chain enzymes. It's just important in general in a cell for helping enzymes do their job. And so if we break down that many mitochondria, maybe it's releasing iron. The problem is iron accumulation is bad for cells because it can interact with things like hydrogen peroxide to make our nemesis reactive oxygen species, cause lipid peroxidation, and basically lead to something we like to call ferroptosis or iron-dependent cell death. Kind of a recently discovered pathway that, of course, as all pathways, is now implicated in like 12 billion human diseases. We're starting to realize how important iron is in a cell, and more importantly, how important it is to maintain iron levels at a regular level so that they don't accumulate and become hazardous. And so how can you, and just as an add-on, I didn't show all this data because it's a lot, but we actually see iron decrease in the muscle. This is in crickets. So we see iron go down quite a bit. And between these two time points, it's going down in the muscle and leaving the muscle and accumulating in the blood. So we know that iron is actually probably causing this color change. And because it should release ferroptosis, we were then thinking, but it doesn't, because some crickets appear to maintain some functional muscle, at least in the VM. And I'll reintroduce the beetles and remind you that they don't undergo apoptosis, and they even have to regrow their muscle after. So, all of this iron and lipid peroxidation could be bad, but apparently it's not. And so that led us to think, well, maybe they're really good at sequestering iron with iron binding proteins like ferritin and transferrin. Yeah, insects have those too. Or maybe they're really good at dealing with the lipid peroxidation. They can increase their antioxidant capacity so that you never really get to this point. And this is kind of just what my lab and my collaborators are starting to work on. And we have like a little bit of preliminary data to show that crickets and beetles do appear to increase iron transport. So if we take a look at the beetles here, we have our same kind of day-long time course of their muscle breakdown. And we see they increase transferrin and ferritin in the DLM, which is really interesting. As normally in insects, we think of these iron transport proteins only being in their blood, but we see a lot in their actual muscle. In the beetles, this is ongoing work. One of my undergrads, Alexandra, she's made ferroptosis her personality and she loves it and is gonna be the one to discover how beetles are resistant to it. And we see ferritin goes up a little in diapause, but this work is still ongoing. But the nice thing is she does have a knockdown working for ferritin, so stay tuned. And this transport of iron could then go and just prevent lipid peroxidation, right? And so we've done a little bit of work in the crickets. This is kind of a messy, casual Western blot staining for 4 H&E protein adducts, which come up in response to lipid peroxidation. We don't see much difference in the DLM. Again, this is ongoing work. And we haven't done any of this yet in the crickets, or sorry, in the beetles, so stay tuned. But I think the cool thing is we now have this comparative model for potential ferroptosis resistance, which we're hoping people in the human world care about, because we certainly are not resistant to ferroptosis. So what we think is happening in the crickets is that these degrading or histolizing muscle cells are releasing iron, but that's being picked up by proteins like transferrin. And the cool thing about transferrin is in insects, in their developing ovaries, there's a lot of proteins that can bind to the cell surface of an oocyte and be taken up and used in development, and transferrin is one of those proteins. And then, of course, iron can then be used to make mitochondria or help with whatever needs to be done for development. The beetles are a little bit different because the cells could be releasing iron, but instead of dropping it off at the ovaries, which are not developed, there may be sequestering it. Maybe it's hanging out in the blood all winter, so that by the time they need to regrow their muscle, they have all that iron stored, ready to go. And so we're in the process of testing these, trying to get funding to test these two hypotheses, and I'm really excited about this. I don't know if anyone else is, but I am. Okay, so I'll kind of end off and just bring it back to the big picture a little bit. The beetles in winter can break down and regrow all their muscle, which is really cool. The crickets can break down one muscle type but maintain the other. So still, it's so cool in my mind, maybe less cool. The beetles do this to save energy to endure these periods of low resources when they're not eating. Whereas the crickets are doing this to reallocate energy for reproduction. And I think it's a really cool model to understand how this adaptive flight muscle remodeling, especially with autophagy, has evolved in response to energetic stress. And in the context of these bugs and these really important life history transitions. So in the context of a beetle, between being winter dormant and spring active, or if you're a cricket, going from being flight capable to reproductive. And I won't leave up on bugs because I know you guys care about humans. This is the end goal, to try and convince NIH and CIHR to give me money to turn these into therapies so humans can regrow their muscle without exercise. But in all seriousness, if we take age-related muscle atrophy, for example, and this is in the context of sarcopenia or anything else, we see a lot of hallmarks of muscle aging, right? One of them is actually impaired autophagy, mitochondrial dysfunction. One of them is iron accumulation and risk of ferroptosis, and of course, reduced muscle mass, things like reduced myokine signaling. Beetles and crickets have enhanced autophagy and mitochondrial quality control. Crickets and probably beetles can sequester iron and avoid ferroptosis under seemingly atrophying conditions. And of course, these beetles can regenerate this extremely atrophied muscle, so they might actually be really interesting models that we can use to better understand muscle in our own bodies. So I think I'll end there. Thanks so much for listening. And yeah, I'm happy to chat more and answer any questions. Thank you.
With that, the floor is open for questions, and we have our first one come up.
Can I ask some super basic questions?
Absolutely.
So when you say the muscle looks red, pink, or white, is that what we lay people would just say dark beat versus light beat?
Good question. No, because the muscle fiber types are a little bit different in insects. All of the muscle should be like, I guess, it should be more like aerobic and like oxidative fiber-like. So there's lots of mitochondria. And I guess to some extent, but if there's no mitochondria, it's no longer oxidative. So it could be, but I guess my point is we don't classify red and white, like there's never really a point in a normal insect not degrading muscle where white muscle exists. Does that make sense?
Yeah.
Okay.
Okay, so I have a question about crickets versus beetles, because at one point you said, oh, these cricket muscles look a lot like the overwintering beetle muscles, but to me they didn't look this similar, because it looks like these have pathetic mitochondria, whereas the beetles had broken down mitochondria.
Yeah.
So isn't that a kind of a big deal, or is it kind of?
Yeah, no, that's great. And I think, yes, so it is, and it's interesting. And I will say one thing that we caught there was interesting is mitochondria are really close to the nuclei, which is one cool thing. In terms of the beetles, we were looking at a longer time scale, and what we're trying to do in my lab is kind of narrow that time scale down. So there's two things that could be happening. The crickets might not have gotten to the point of such mitochondrial depletion. Or the crickets are actually maintaining the really small pathetic mitochondria to a greater extent, which I agree with you, is interesting in itself. So it's I guess, a timing technical thing that we need to sort out, but yeah, that's really interesting.
So the breaking down the mitochondria is a big surprise to me, not knowing this field at all. Because I thought, again, for lay people, we hear that mitochondria are parasites or eukaryotes. And so I'm kind of surprised that you can break them down, because then isn't the DNA just kind of mixed up with the, parent cell? And so then how are you going to get back mitochondria again if you just stuck it all in a blender?
Right, and so that's why I think we're not capturing it on the micrographs, but I do think the beetles keep some around. Because you're right, mitochondria come from mitochondria. You need both the mitochondrial genome and the nuclear genome to make mitochondria. So you're right, I would assume that when the mitochondria are broken down, that DNA is also degraded, probably through similar processes. So by the time the mitochondria are broken down, that DNA is now just nucleotized and gone. So I assume that there are some remaining which help the regrowth process. And that's something specific we're starting to look at in the lab.
And so last thing, I think it's great that you mentioned sarcopenia, and I have a question about that, which is, so supposedly with older adults, when they have muscle wastage, they can exercise and grow back muscle, but apparently it's really hard to get back fast twitch muscle. So then, is there a gene that needs to be expressed, or a protein they need, or what would be the magic thing that would let them grow back fast twitch?
Yeah, that's a great question. Interestingly, what I think is happening in the beetles is they're using this minimal glycolytic capacity they have in their cells to basically power the growth of a few mitochondria to then get up the oxidative capacity to, I guess, act more like a slow oxidative muscle. So if we can find out what that cue is, then it would be really interesting to think about how we can translate that to like growing back muscle types for sure. Yeah, again, I teach muscle physiology and bioscience, and most of what I teach is vertebrates, and I don't even tell anyone that the insects are like completely wrong and they don't have like the same fiber types. It's mostly the flight muscle itself would be analogous to slow oxidative, but it also has like glycolytic capacity. So it's, yeah. Anyways, that's my roundabout way of saying yes. I think we could maybe think about that. Yeah. Good questions.
Other questions? So while we're waiting for one, I did have a few that are kind of naive questions, but
nope, nothing.
And you sort of answered them, but just to confirm, do crickets then not have their flying muscles after they are pregnant and?
Yeah, so they just lose it. It's like use it and then lose it. Because ideally, once they found a mate, the females can store a lot of sperm and they can lay a bunch of eggs. And so there's not really any need to fly around to find someone new. Or at least the costs are greater than what would be beneficial to keep flying around.
And then what is a cricket's lifespan?
Yeah, so in the lab, a long time because they're living in paradise with all the food and water they could ever want. I would say about, I don't know, maybe a month or two. In the wild, probably much shorter through like predation. There's like less access to food and stuff. So in the wild, I would probably give it a few weeks. So in the scheme of things, I guess I didn't really emphasize that the muscle breakdown usually happens between 5 and 7 days. So that's like a third of its life. So yeah, it's a kind of a long process for them.
Makes sense. Yeah, that's where my question came from, because your x-axis was like 3, 5, 7 days, and that's how long these things live.
Yeah, and we're in the process of trying to, so minute and I need for a week. I wasn't allowed to be on it. Thank you, Erica. To use them as a model for exactly this. And so we have some ongoing work there to see what happens in the like old wizened crickets to see how far they can maintain that muscle into their life. Because it looks like the longitudinal muscle that is broken down, that's broken down really quickly, and as they age, it wisps down to almost nothing. So unlike the beetle, they do completely degrade the longitudinal muscle, but we're almost in the process of figuring out how long we maintain the other one. Good question.
Do you think there's any functional differences between the muscle in animals that go through in like vertebrates that go through the diapause versus not? Like, are they sacrificing some kind of function in the mitochondria or in the myofibrils or whatever?
Yeah. That's a good question. So I guess, do you mean like after the fact, like once they're out and they've gone through that process?
I guess both. Like,
Mhm.
would there be some difference in the function of the muscle in whether they go through diapause or?
Yeah, that's something I'm really interested in. I'm interested in doing more on like the emerge side of things. Like once they've grown back their muscle, what does that mean for the muscle function? That's a good question. I think that probably, yeah, not that much, because the idea is once, at least in these beetles and a lot of other similar ones, when they come out of diapause, they need to be able to fly to find food and a mate. And so like adaptively, I would hope that it would be selected for like even optimized muscle function. If you even look at, if you remember, what I think is really interesting, I'm not going to reverse back to the whole beginning, that's a lot. But mitochondrial oxygen consumption goes down during diapause in the muscle and it goes back up. And it's actually higher after diapause than it is before. So that, I guess maybe that's a little bit of evidence that it could be better, if anything. Yeah, that's interesting.
And then in terms of kind of maybe answer this a bit before about the fiber types.
Mhm.
So then in the animals that are, say, the crickets, where they're still in, they're not totally in hibernation, like they're in
Yeah. Mhm.
Is there kind of a transition in fiber type, or like in terms of they're then using anaerobic metabolism in those muscles when they do use them?
Yeah, we're trying to figure that out right now. So they have a seahorse. They're working on it. I would guess yes, especially the ventral muscles that are maintaining, because they are degrading most mitochondria, keeping some around. So I would assume that they start to shift more towards like glycolytic capacity for sure.
Okay, we had a question from Walter and then Ariana.
Yeah.
Hi.
Thanks, interesting talk. At the beginning, when you talked about the beetle, you said that you use the diapause simply by changing the light exposure.
Mhm.
So does it not depend on temperature at all? It just makes the day shorter if you go into hibernation.
Yeah, so we do bring them down to 15 as more of a realistic fall simulation, but we don't need to. So basically the diapause is cued at 24. When we raise them as from eggs to adults, it's 24 degrees Celsius and a short photo period, and that makes them go into diapause.
And then you make all these measurements that you have made in the laboratory, I assuming normal temperature. And I was wondering, do you think those processes and signaling pathways are affected when it's minus 30 degrees compared to the plus 20?
Absolutely, Miranda can tell you all about that. She's sitting right there. That's her master's to some extent. But yeah, so there are a few things when they're underground. If it's minus 30 outside, right now, actually, what is it, minus 15 today? They're probably very cold because there's not a lot of snow cover. When there's snow cover, if it's minus 20 outside, they're probably only experiencing about minus 5, minus 10. And so the answer is yes, temperature probably does a lot. We're actually in the process of starting to look at the temperature sensitivity of these processes. So what we think, at least what we hope is that if we lower the temperature, that it just prolongs the process. That everything happens, it just happens more slowly. Yeah, so that's the idea.
So about 10 years ago, people started complaining in Calgary and in the mountains that it doesn't get cold enough anymore.
Yep.
And it doesn't kill the bark beetle.
Yep.
So I guess it seems that certain animals, certain insects will die off when it gets too cold, but they can
Yep.
survive when it's only minus 15 or
Totally, yeah, so that's like the second half of my research program, which I didn't think anyone would be interested in hearing about today. These beetles, their kind of lower lethal temperature is about minus 13 when they're like acclimated to the cold. And yeah, the bark beetle can survive down to like minus 50 or something ridiculous. And on top of that, the bark beetles are living inside trees, and that's actually quite thermally insulated. So like, that's why I say like they're all around the insects, but they're not actually experiencing the same temperatures as us because they found little microhabitats underground in the trees, various places where they're able to buffer themselves from the cold. But then on top of that, they've evolved like biochemical and cellular strategies to be able to protect against the severe cold.
And then if I understood you correctly, in the first part of your talk, you talked about metabolic cost, oxygen uptake of the entire animal. So how do you do that?
Yeah, so we don't have it yet. We will in August. It's a flow through respirometry setup where we'll have like activity chambers for the insects that's then connected to, we run like CO2-free air through. And then a CO2 sensor then can detect when the insects are breathing out carbon dioxide, and we can just convert that into a rate. So the caveat is we can only measure carbon dioxide production. I didn't have enough money to buy the oxygen sensor, so not yet we will be able to actually measure oxygen consumption, but we use CO2 production as a proxy.
Then about the selective adaptation of the two muscle types in your cricket. At one point we did a little bit of work on Drosophila, and we know from that indirect leg muscle Drosophila is very, very different than the leg muscle. And so you also have structural differences between your two muscles, the one that help in the walking and the other ones that are just there for flight.
That's a really good question. I think there's a paper from like the 80s that did like a deep dive into the ultrastructure. And I don't think there's any appreciable differences until they start to diverge in degradation. And then it's mostly the mitochondria and the protein in terms of ultrastructure. But I don't know about anything else. That's a very good question. There's definitely more of the ventral muscle.
And so in these beads, they're also stretch activated.
Yeah,
like in
It's the same.
But the leg muscles, the ones that help in walking, they would also be stretch activated.
I think so, yeah. We're actually starting this summer. We're gonna do all of this, well, not all of this, but we're gonna do some of this in the leg muscle of the beetles and compare. And we think that they're maintaining the leg muscle because it's much more pathetic muscle. There's not as many mitochondria. It would be more analogous to like whiter muscle, I guess. So we're thinking that there's no reason to degrade that muscle, so they keep it around, especially similar to the crickets. Even though they're underground, they need to be able to walk when they kind of come out and occasionally like move around to get away from predators and stuff. So we think that the leg muscle is being maintained, and if we figure that out, then we can use it as a nice internal control. Yeah.
So actually, it reminds me of another question that I had. So when the bark beetle wakes up or comes out of the pond, can he immediately fly? What does it take?
That's a good question. I will say the bark beetle does break down its leg muscle. It's one of the, and all of these papers are from entomologists from like the 60s and the 70s that just pick up a beetle from nature and say, oh yeah, slight muscle degrades. And I said, why is that not cooler? Like people didn't really dig into that. But we don't know much more about the bark beetle because they're very hard to study because you need a quarantine facility to be able to bring them into the lab. So I don't do any of that work. I have a colleague at U of A that does it, but they can only bring the beetles in dead. So yeah, it's hard to do that kind of fine-tuning work with them.
The last question, if I understood correctly, at the end you mentioned that you get an apparatus for measuring ATP consumption, oxygen consumption of single cells.
So yes. The one that I use mostly does it in isolated mitochondria and permeabilized muscle. So it's not like cultured cells, but you can just literally dissect out a muscle and permeabilize it with like saponin or something mechanical. And then, or you can isolate mitochondria and measure oxygen consumption. Does anyone here have a seahorse? Does anyone know what that is? So the other machine, which we might be getting in our department if we get the RTI, that measures ATP production and consumption, oxygen consumption in cells, in cultured cells.
In cells.
Yeah.
And also in isolated muscle fibers.
Yeah, yes. It's a little trickier in that one when it's not an isolated cell, but yes, to all of that, to some extent.
And what time resolution does it have?
So
Does it measure it essentially as you vary force, you can measure the ATP.
so it's all metabolic. So at least, so for example, in the one that I've used and that we are getting in the lab, it's, if anyone's interested, the company is called Ouroboros in Austria, and they make these, and they're very popular. So you isolate mitochondria, and they have two chambers that have oxygen electrodes to measure oxygen concentration, and then they're kind of semi-sealed to the environment. So you can, put a bunch of oxygen in there, know how much there is, put the mitochondria in, you plug it up so it's mostly sealed, and you actually feed it metabolite substrates. So the isolated mitochondria, although it's isolated, it'll still work for a set number of, hours to a whole day, even if you're really good at it. For example, like what do mitochondria need? Mitochondria 101, I need ADP to make ATP, so you give it some ADP. It needs, let's say, pyruvate malate succinate, so you give it that. You can give it inhibitors to inhibit different parts of the electron transport to assess function. It's really fun. Yeah.
Thank you.
Hey, Ariana had a question.
So I was just wondering, like for breathing holes, like what happens to them when your muscle atrophies? And then if like the insect breathes easier when the muscle isn't like contracting?
Yeah, so his question was about how, I guess really at its base, how insects breathe. So I'll maybe go into a little bit more detail. They're called trachea. So if you think of like, I'm a bug, inside of my body, there's gonna be a bunch of really tiny microscopic holes. And you go into those holes and there's almost like a network, almost if you were to picture like the network of like lung bronchioles in a human, that's kind of like tubes that go from really big to really small terminal endings that poke right into the cells. And so that's why insects are such good athletes per se, and that's why they can fly and we can't, is because the oxygen delivery is super efficient. We have to breathe it in, it has to go into our lungs, into our bloodstream, finally into the cell. Insects, they're always, you know, ventilating and oxygen's always getting into their cells. So your question was about if the muscle's not contracting, if there's less oxygen.
Or like the holes are like open?
Yeah, so the holes are always open inside the body. Outside the body, there's a musculature outside that opens and closes them. So their breathing is kind of always opening and closing this. Opening to let oxygen in, they close to basically prevent water loss, and then they open again, and then CO2 can be let out.
And then when the muscle atrophies, they don't, they just stay.
Yeah, so it looks like, and this is something we need to maybe do a deeper dive in, but it looks like the terminal endings kind of like stay in the cells, because at least in the beetles, the cells are still there. They're just like pathetic shells of cells. And so they're still getting oxygen because they still, to some extent, you know, even though there's not a lot of mitochondria, as you know, someone asked before, there's probably like a few. They still need a little bit of oxygen, but it would be cool to maybe see if their cells can survive under like more hypoxic conditions than others. But I guess it's basically like chemistry, physics. Like if the oxygen can get in, it's always going to go in, right? Mm-hmm. Yeah.
Other questions from the audience?
Art, yeah.
Okay, I don't feel bad about asking this because no one is asking, but how does a caterpillar become a butterfly? Is it like the beetle where it's breaking down everything?
Yeah, so that's an interesting question. So, good observation. In metamorphosis of insect life stage, how do insects work? They hatch from eggs. A lot of them go from larva to larva. If you're a butterfly, you grow up as a caterpillar. In between each of those stages, when they undergo metamorphosis, there's mass breakdown of muscle and regrowth. So that's actually not novel. We've known that about insects for a long time. And so it's not, there's more apoptosis is the answer. It's not the same. It's full breakdown and digestion so that they can grow new muscle and new body parts, but it's not the same as we're seeing with beetles, if that makes sense. Is that enough detail? And then they form a cocoon, and then they
Okay, any other last questions for Dr. Lebenzon? Oh. All right, then let's thank her for talking.